Good morning, students. This is Dr. K Surendirakumar, Assistant Professor, Department of Microbiology, JJ College of Arts and Science, Pudukottai. You already know about plant tissue culture and plant tissue culture techniques and how it will be help to develop new resistant plant varieties like hybrids. So here we are going to discuss the another plant tissue culture technique is called protoplast culture. Before going into the topic, we need to understand what is plant cell and what is protoplast. Plant cell, it have outer thick wall, then inner thin plasma membrane and other cell organelles. When we look into it, protoplast, the plant cell without cell wall is called protoplast. It means the protoplast outer membrane is plasma membrane and other cell organelles are same. So this is the difference between plant cell as well as protoplast. The term protoplast was coined by scientist Einstein in the year of 1880. He referred the protoplast means the naked plant cell. Then later in the year of 1892, Lurker, he successfully isolated the protoplast for the first time using mechanical method. Then what is the purpose of protoplast and protoplast culture? This protoplast culture is a unique experimental systems for studying structure and functions of the plant cells. Then it will be help to study the plant genetic transformation and cell modification studies. Even it will help to study about plant virus infections. Finally, it will help to introduce the genes of agriculture and horticulture crops for getting hybrids. In this slide we are looking the left side plant cell. Generally the plant cell, the cell wall is made up of pectin or cellulose. That's why the plant cell outer coat is so thick. So if we need to isolate the protoplast, what we will do? We need to remove or digest or cleave the plant cell wall by mechanical method or enzymatic methods. Mechanical method means simply we need to cut that plant cell wall and remove the protoplast. In case of enzymatic method, we need to use enzymes. These enzymes digest the plant cell wall and release the protoplast. So here in the right side we are looking protoplast. It have without cell wall. So the outer membrane is plasma membrane. Two different concepts is there. One is spheroplast, another protoplast. When the plant cell wall is partially removed, is called spheroplast. When the cell wall is completely removed, is called protoplast. So in this slide we are looking how the protoplast and protoplast culture is evolved. In the year of 1892, Lurker, who isolated the protoplast for first time using mechanical method. In the year of 1960, Cocking, who also isolated the protoplast from tomato root tips using the enzyme cellulase. Then in the year of 1968, Takebe and his co-workers also isolated the protoplast using enzymes. In the year 1974, Cow and Michaluk was proposed PEG for fusion of protoplast. What is PEG? PEG means polyethylene glycol. After isolation of the protoplast, if we need to fuse this protoplast and produce the hybrids, then finally in the year of 1979, labor who successfully fused two different tobacco protoplasts to produce that hybrid plants. In protoplast cultures, we have three different stages. First one is isolation of protoplast. Second one is protoplast fusion. Third one is development of regenerated plants from the fusion of protoplast. It is called generally hybrid. So these are the steps. Is mainly depend on what kind of plant genotypes we are taking up, what kind of species we are selected, which part of plant material we are used, what kind of enzymes we use to isolate the protoplast, and what kind of culture conditions we provided to grow the protoplast. So these are the main things. Once the isolation of protoplast is happened, the plant cell, when we transfer into the suitable media, it will regenerate their cell wall within 24 hours. Later, the cells will multiply and generate undifferentiated mass of cells is called callus. From this callus, we can carry out two different organogenesis. One is embryogenesis. Second one is organogenesis. Embryogenesis means after callus culture, we are going to suspension culture and we produce embryos. From that embryos, we can produce plantlet. Then another one is organogenesis. After the generation of the callus, we can directly generate shoot or root. Using different medias, plant growth hormones, and the culture conditions. The first one, isolation of protoplast. How we can isolate the protoplast? Two main processes there. One is mechanical method, another one is enzymatic method. Mechanical method means simply cleave the plant cell wall and release the protoplast by means of cutting the cell wall using knife and grinding the tissues using mortar and pestles. So what will happen? In that mechanical method, we will get minimal amount of protoplast. Why? We are trying to rupture the plant cell wall, isn't right? So when we rupture the cell wall, what will happen? Due to our giving pressure, protoplasts also will be broken down. So the yield of the protoplast by mechanical method is less, and it have certain disadvantages. One is lower protoplastic yield. Second, labor intensive. Third one is the viability of the protoplast is low. There are another alternative method is enzymatic method. Here we simply cut the plant segments and add our enzymes, whether cellulose or hemicellulose or pectinase, whatever it is. Then we need to maintain certain conditions of digestions, because enzymes is more sensitive on pH and their temperature and their incubation period. Three different things is there. Generally, this type of enzymes is more active at 4.7 to 6 pH. Temperature should be 25 to 30 degrees Celsius, and the incubation minimum 6 to 12 hours. Then automatically the cell wall will be ruptured by enzymatic way, and the protoplast will be harvested by centrifugation and purification. Then we can go for further cultures. In this slide we are taking leaf as well as callus. We can isolate the protoplast from the leaf or other undifferentiated mass of cells like callus. So here what they are doing is simply cutted their segments and dip into that enzymes for 6 to 18 hours at 25 to 30 degrees Celsius. Then protoplast will be, the cell wall will be digested, the protoplast will be settled down, then those protoplast is transferred to centrifuge tube. Those centrifuge tubes we need to be centrifuged. So there what will be happen? The cell debris means the plant cell wall will be settled down and the less weight protoplast will be go up. Then those protoplasts will be washed and they will be suspended or transferred to other media for culture of their protoplast. The second one is protoplast culture. Generally the protoplast culture having 4 steps. First one is selection of the plant material. In which plant, which segments we are choosing is more appropriate to get enough quantity of protoplast. After selecting the plant material, we need to be surface sterilized using 70% ethanol for avoiding disinfectants. Then later we will go for protoplast isolation by means of chemical or by means of mechanical or enzymatic method. After that, the protoplast should be cultured using appropriate culture medium like MS media. When we inoculated into that media, the protoplast develop the cell wall and followed by cell division. It will form small colonies, then it will be go for callus. With that callus we can go whether embryogenesis or organogenesis. Then finally the desirable plant regeneration will be happen. During the protoplast culture, okay, in this slide we are taking 2 different explants, explant A and explant B. Both the explants we are taking leaf and cutted to small segments and digested with enzymes. After isolation of the protoplast, what will happen? We have fusion. Generally the protoplast fusions happen by polyethylene glycol, and after the fusion we will generate heterokaryons. Here the protoplast is vegetative cells, 2 explant is there, okay, explant A, explant B, so both having protoplast 1, protoplast 2. If we fuse together 2 protoplast, what will happen? 2 possibilities is there. One is exchange of genetic material, it means form heterokaryons, is called hybrid. 1 plasmid, 2 genetic material is called hybrid. That another one, when we fuse together, only 1 genetic material will be prevailed inside the protoplast, is called hybrid. So 2 type of protoplasmic fusion and the result is there, that is there. Then next we will make a callus. From there we need to be regenerated, whether it is through embryogenesis or organogenesis. Here the protoplast fusion, I already told now, plant A and B, protoplast is there, polyethylene glycol is helped to fuse their protoplast. If the fused cells having 2 nucleus, plant A and plant B nucleus is called heterokaryon, and these 2 fuse together and 2 characters will be exchanged, is called hybrid. The hybrid, why we need to be produce hybrid plants? For getting more yield, to generate resistant varieties. whether any pathogens or stresses, biotic and abiotic stress combinations. Then another one is hybrid. The hybrid means when we cross two genetic material, only one genetic material will be prevailed, is called hybrid. Okay. Among different stages of the plants we are selected, different factors, how the protoplast will be successful. This successful protoplast culture is mainly depend on plant source. What kind of plants we choose, what is the genotypes, what is the age of the plant material, how the growth condition is there, then how the surface sterilization we done it, and what enzymes we choose to digest the cell wall and isolate the protoplast, how we purified the protoplast, whether we select, when we culture the protoplast, whether the protoplast is viable or not, then what the culture medium we used to grow the protoplast, and what kind of growth hormones we used, and what is the growth conditions, temperature, light and cell density. So all the things is will decide the successful protoplast culture techniques. Then what is the uses of isolated protoplast culture? This will help to study the biochemical and metabolic pathways of the cells and fusion of two different somatic cells. I already told protoplast is a somatic cell. Just got it? Okay. The fusion of two different somatic cells to get a somatihybrid. Then production of hybrid and genetic manipulation. It means exchange of genetic material from one plant protoplast to another plant. Then drug sensitivity. Okay. Thank you. Today